Reducing dependence is a major research avenue within mu opioid receptor signaling. For a future in which opioids could be used as long-term mood enhancers, pain relievers, anxiolytics, and antidepressants, we would need to ensure that the development of dependence was substantially limited. Generally, dependence manifests as drug craving, drug seeking, and, perhaps most importantly, withdrawal symptoms. These can include sweating, high heart rate, nausea, constipation, shivering, and generally can be very unpleasant for the person experiencing them. So much so that they fail to break out of the addictive routine that they might find themselves in. Central to dependence has been the AC CAMP PKA pathway. This pathway begins with AC, adenylyl cyclase. Adenylyl cyclase is an enzyme which catalyzes, that is, drives or speeds up the conversion of ATP into CAMP. CAMP is a major secondary messenger. Secondary messenger meaning an agent that carries the signal inside the cell where the initial initiator of the signal cannot enter. So in this case, the more agonist acting with the more receptor, instead of leading to direct effects by interacting with agents within the cell directly, confers or transduces its signal via CAMP, which can interact with more of the effector proteins, enzymes, and kinases within the cell. So CAMP interacts with a variety of intracellular agents, but PKA, or protein kinase A, stands as one of the major downstream kinases that mediates the effects of CAMP activation. In acute opioid exposure, the ACCAMP PKA pathway is inhibited. This is mediated through the G protein A subunit. In chronic exposure, however, we have found that the G protein A interaction with AC actually becomes stimulatory. This is of most importance to our purpose here to reduce dependence because dependence occurs over long-term chronic exposure. Therefore, interventions aiming to reduce this long-term increase in activity have been explored to potentially reduce dependence. For example, AC inhibitors have recently been explored in preclinical models and have been found to reduce withdrawal-like behaviors and other dependent-like behaviors like drug seeking and drug craving. Elsewhere, PKA inhibitors also hold promise. But here, the literature tends to use or mention PKA inhibitors mainly as tools to identify and elucidate the functions of protein kinase A itself. Some have argued that these direct agents acting on the PKA CMPAC pathway won't be ideal because of their off-target effects. To elaborate, the ACCAMP PKA pathway is involved in several different intracellular signaling cascades. In fact, a whole range of agents acting at the cell surface membrane use this pathway to mediate intracellular effects. Therefore, it is a worry that while agents acting directly on this pathway may be able to reduce dependence, they may also introduce adverse physiological effects or disrupt other physiological signaling cascades going on elsewhere, which could ultimately manifest as some other symptom or disease that we wouldn't want to induce on someone. Within a brainstem region called the locus coeruleus. The overactivity of the ACE-CAMP-PKA pathway leads to the specific effect of enhanced noradrenaline release. This is thought to happen because PKA, through the mediation of other downstream signaling molecules, increases the overall firing rate of the neurons in the locus coeruleus. Because neurons in this area are noradrenergic, in other words, because they produce noradrenaline, this increased activity translates to a greater efflux of the hormone or neurotransmitter, noradrenaline. Noradrenaline is known to cause some but not all of withdrawal symptoms, including those associated with higher metabolic activity such as sweating, high heart rate, shivering. And so agents aiming to prevent this enhanced release of noradrenaline have been um, have been trialed to try and prevent these noradrenergic withdrawal symptoms. Clonidine and lofexidine are two examples of agents that prevent this output of noradrenaline by blocking the effects of PKA. They don't do this directly, but do it via the introduction of adrenergic receptors at the cell surface, which encourage the uh, movement of noradrenaline into the cell. So effectively, these receptors sequester noradrenaline within the cell and prevent their efflux, and in doing so, prevent the noradrenergic withdrawal symptoms. Clonidine and lofexidine are actually um, FDA approved in some indications, and a number of clinical trials have shown them to be effective to prevent or reduce withdrawal in opioid use disorder. Um, their use as adjuncts to opioid prescription for chronic pain, for example, is within clinical trials as well. So there's a promise with these agents. Rather than directly targeting the downstream components of this pathway, the idea of preventing this transition between acute inhibition of AC to chronic activation of AC might stem as a more promising and more selective, that is, intervention to prevent the development of dependence more upstream. One agent that has been shown to achieve this is sarcatinib. Sarcatinib is an inhibitor of SRC kinase, which has been shown to introduce a phosphorylation of the mu receptor at residue tyrosine 336, which ultimately seems to drive the transition of G protein A as an AC inhibitor to G protein A as an AC stimulator. Therefore, by blocking this SRC kinase, it has been hypothesized and shown in results that we can prevent this compensatory stimulation effect that has been seen from G protein A after chronic exposure, therefore retaining the inhibition of AC, preventing the overactivity of the AC-CAMP PKA pathway, and reducing withdrawal and dependence. Evidence shows that sarcatinib can achieve this in preclinical models. More so, sarcatinib is used for other indications as an approved therapy, so it is known it's safe and well tolerated in humans. Currently, clinical trials to assess its usefulness within opioid use disorder or preventing dependence ever developing for those on chronic prescriptions is being investigated in humans.